VSL Test Interpretation, the Standard Plate Count, from the Cornell University Milk Quality Improvement Program. In this training, we're going to discuss the importance of the standard plate count, which is abbreviated SPC, the background of this test, how it's performed, and how the results of the test are interpreted. The standard plate count is important because, in the United States, the Pasteurized Milk Ordinance, which is the regulation that sets the quality standards for many fresh dairy products, says that milk can have an SPC of no greater than 20,000 bacterial cells per milliliter of milk. High numbers of bacterial cells in pasteurized milk may lead to reduced quality over shelf life. The standard plate count is sometimes also called a total bacterial count or an aerobic plate count, but both of these other terms generally refer to the same test as the SPC. The SPC counts the number of bacteria in a milk sample, and these bacteria could be from the raw milk or they could be from the dairy plant environment. Some of the bacteria counted by the SPC are heat resistant, and that means that they can survive the high temperatures of pasteurization, but some others are killed by the heat of pasteurization, so when we find them, we know that they were probably introduced after the pasteurization step. A lot of these heat-sensitive bacteria can enter through the filler. When these heat-resistant or heat-sensitive bacteria are able to grow in the milk at refrigeration temperatures, their numbers increase enormously over shelf life, leading to high SPCs and to spoilage. To make sure that the SPCs stay low over the milk shelf life, we need to focus on the ways that the bacteria can get into the milk both at the farm and at the dairy plant. There are several approved methods for performing the SPC in the lab. Here at the Milk Quality Improvement Program, we spread a set volume of each milk sample that we collect for the VSL program onto the surface of a petri dish filled with standard methods agar. These dishes are held at 90 degrees Fahrenheit in an incubator for 2 days to allow for the bacteria to grow. Bacteria grow very quickly, and their population can double in refrigerated milk 2 to 3 times every day. Each colony of bacteria that grows on the petri dish grew from a single bacterial cell or colony-forming unit that was in the milk when it was added to the petri dish. Once the colonies have grown, we count them to obtain the SPC value. We count every colony that grows on the petri dish, regardless of what shape, size, or color it is. Now, here's an example of a VSL report. SPC is reported for each test day over shelf life, which is either 14 or 21 days, depending on the plant. The SPC values are in the red box on this report, and they are color-coded based on their value. Values that aren't highlighted are less than 20,000 per milliliter. Values that are highlighted in yellow are greater than 20,000 per milliliter. And values that are highlighted in red are greater than 1,000,000 per milliliter. Looking at the two samples on this report, we can see that there isn't any evidence of bacteria that can grow at refrigeration temperatures in sample 22 because the SPC didn't increase over shelf life. In sample 25, we can see that there was evidence of bacteria that can grow at refrigeration temperatures as the SPC increased to very high levels over shelf life, exceeding the limit in the PMO by day 7. Thank you so much for watching this training. If you're interested in taking classes with us or learning more about our research or becoming a member of our voluntary shelf life program, please refer to the information on this slide or to the links in the video description.